Yeah, bacteriophage time. All right, there's a series of videos that I'm not going to play so we don't get copyright struck, whatever. But they are going to show you a lot of things. So let's go through what you're going to see in these. And these are all short videos. They're like 1-2 minutes each, and they have really cool visuals. They're not cartoons. I think all these are molecular animations of some sort, which is much better, really. So the way a bacteriophage, so this is the most common sort of appearance of a bacteriophage, is something like this. This is a glass model by an artist, but actually it's got a lot to recommend it. It's pretty virus-like, yeah, phage-like. So first thing that happens inside of the infected bacterium, and they get blown up not by lasers but by the virus going inside, but whatever, we'll get to that. The head is going to assemble first, so that's this part right here. And it's made of a bunch of little hexagonal plates, but they actually form on it makes like a skeleton. It's like a cage, and it puts the little plates on the outside to assemble this thing. So we're going to assemble that, and then we're going to load the DNA inside. And so you will see the third video is going to show you the loader, which is a little protein that grabs the DNA and it pulls it up and pulls it up, pulls it up, pulls it up, pulls it up. And it's going to bind ATP in order to do that. You see a little bit of that in the first video and a nice close-up of a different version of this protein in the third video, which is very accurate, and it shows you where the ATP is and everything. While these are happening, the tail part is going to assemble. And so the tail is going to store conformational energy. So it's a long protein, and it could rotate and become a short protein, or it could be like a spring, basically. So the spring can be pulled out, in which case it's going to try to collapse as soon as something changes. There's going to be a triggering event that's going to move the position of some proteins, and it's just like when you have those horrible children's toys, a Jack in the Box. You turn the little crank and it plays the music, all around the mulberry bush, whatever. And when you get to Pop Goes the Weasel, a horrible little clown demon on a spring comes out and startles children. Yeah, apparently parents find this endlessly funny. But it's the same sort of deal. You're going to have stored energy in the form of a stretched spring, and then what happens is that spring is going to collapse. And when it collapses, there's a long rod inside of it, and that's going to be injected into the bacterium, and then the DNA is going to come out. So you can see the DNA injection here. That's going to be based on structures, but it's more of a cartoony sort of way of looking at it. But there are some bacteria that have very long walls or like multiple membranes to get through. And so for those, we're actually going to inject an enzyme called a lysozyme, or an endolysin is another name for these. And those will actually go in and they'll start to dissolve. They'll break apart the bonds that are holding the bacterial cell wall together. And that then allows the syringe to go in further, and the syringe will then deposit the DNA directly into the bacterium. And then you can actually see in the last video, I think that's the coolest one, it's actual photomicrographs of E. coli cells with some virus swarming around. You can't see the virus, but you can watch over a couple of seconds as the E. coli go pop, 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 pop. Yeah, it's just perfect. And that is what happens. The E. coli fill up with virus cells, and the whole thing just bursts and releases a whole bunch of virus that'll then go and hit other cells. Yeah. So look out for those things. All right. Now let's talk a little more about phage and the origin of phage and the origin of viruses. So some of these at the end are kind of mind-melting, at least for me. See what you think of them. So in the center of this thing, I love diagrams that are multi-layered and have a lot of complex information in a small space. So here we've got a bacterial family tree. And so these little rings are connected to all these little branches, and these are different types of bacteria in the world. This is the bacteria tree. And they're all related to each other, so we can show them on the same tree, so that's okay. Then in the rings around them, we have viruses that infect those bacteria. Now, a lot of viruses are not yet discovered or named. Basically, for every living kind of cell, there appear to be several kinds of viral parasites that infect it. And the only difference between, viruses that have, or bacteria that have one kind of virus and bacteria that have several kinds of virus are essentially how well they've been studied. So in each of the rings, you have different types of virus that are here. And so you can see that there are some viruses that seem to hit pretty much everything. And there are some viruses where you'll find them in certain groups, like this crazy guy out here on the edge, but not other groups. So these things may be more recent viruses that only infect one group, is the idea. Look over at archaea and you see some of the same things. Now, some of them will be the same as the ones that infect bacteria. They're going to be related viruses infecting different domains of the tree of life. So these little things, they look like a soccer ball on a stick. Those are going to be called tailed phage. They're in a large group called the Caudovircetes. Caudus means tail. They're showing them here as Duplodnaviria. That is the correct name for the larger group that contains them. That's their virus name. But yeah, they would generally be called tailed phage. Okay, and so from this, you can get two different conclusions, I think, that make a lot of sense. One is the one that they make in the paper, which is at the top in red. So their conclusion is that, you know what, we've got certain types of phage that seem to infect everything. They infect both bacteria and archaea pretty broadly. So probably we know bacteria and archaea, or at least we hypothesize that they share a last universal common ancestor. So that's LUCA here. Last universal common ancestor. So deep in evolutionary time, a couple billion years ago, there was something that had two kids and one of them turned into bacteria and one of them turned into archaea after many, many, many long, wonderful journeys. You'll have some viruses that only infect bacteria, and so those may have infected the last bacterial common ancestor. Some that only infect archaea but infect very widely. And then you'll have some that only infect particular archaea and are found in no bacteria. And so the idea is that those have evolved while archaea have existed, and they are archaean specialists or bacterial specialists, if you're going the other way. So that's a neat hypothesis, and there may be some truth in that. Another interpretation would be that since we know viruses evolve much faster than bacteria or archaea, or anything cellular for that matter, it is very possible. And we know that viruses can jump from host to host. Like, look at SARS-CoV-2. That's done that very well. And lots of other viruses. Actually, all the other viruses have done that at some point. So it's also possible that the host range reflects sort of the plasticity of the virus. It reflects the range of things that virus is able to adapt and infect. So there will be some proteins that are actually in common between bacteria and archaea. And if a virus learns to grab onto one of those proteins, for example, then at least theoretically, you could end up with a virus that's able to infect some bacteria and some archaea. Because once the virus gets in, mostly it's going to be copying itself. It's going to need some kind of generic ribosome. But since all ribosomes are related to each other and essentially the same, and since they all run on more or less the same genetic code, there's actually not a lot of optimization that will be needed, even to make as big a jump as a jump between domains, between an archaeal host and a bacterial host. So that's interesting. I guess the truth could also be somewhere in between. You could have some viruses that are ancestral and have co-evolved along with the hosts, and others that jump around a little bit more. And with the right kind of evolutionary biology study, you might be able to tell those apart. So stay tuned. I would imagine over the next few years, we'll get some contenders for answering these questions. All right, so we're going to pause this section, and then we're going to go into the really weird stuff, and the really weird stuff is honestly the best stuff. So we're going to talk about Spiegelman's monster. We're going to talk about Yeah, compartmentalization. We're going to talk about the origin of viruses a little bit. We talk about this again later in the class, but stay tuned for part 3.